All right, good morning, everyone. My name is Liza Lester. I work in AGU Media Relations. This is Ocean Sciences Meeting 2014, the first of our press panels today. Ocean Sciences is actually a collaboration of AGU, the Oceanographic Society, and ASLO. So I want to make sure that they are honored as well. And I'm going to turn this session over to Kate Ramsayer from NASA Goddard Communications in just a moment to introduce our panel. But I wanted to let you know that this panel, we will first have each of our panelists introduce themselves and their expertise, give you a little preview of PACE, and then we're going to go straight to Q&A. We want to reserve most of the time for Q&A, so I hope you will ask lots of questions. And we also have a few people online, so if you're online, please put your question in the Q&A box, and we can either ask that question for you to the room, or we can open your mic and you can ask it yourself. And I think that's all. I'm going to turn it over to Kate.
Thanks, Liza. Welcome to our panel on NASA's newest Earth science satellite, launched just last week on February 8th. It was a beautiful launch. We were so excited to be there, and there's no rest for the weary. We're now ready to turn this thing on, get some data, and put it to use. So we wanted to put together a panel of some of the folks who are going to dive right in to the PACE data, and I'll turn it over to them. Our speakers today are Jeremy Wardell, who's our PACE project scientist from NASA Goddard, Clarissa Anderson, and I'm going to say these organizations, the director of the Southern California Coastal Ocean Observing System, and the director of the Cooperative Institute for Marine, Earth, and Atmospheric Systems at UC San Diego's Scripps Institution of Oceanography. Shelley Tomlinson, a research oceanographer with NOAA's National Centers for Coastal Ocean Science, and Amy Neely, PACE support scientist and ISAT-2 applications lead at NASA Goddard. So with that, I'll turn it over to Jeremy.
Thank you for coming. Do I need the mic closer to me, or is everything? We're fine? Okay. Thank you for coming. As Kate mentioned, I am the project scientist for PACE. I'm going to be enthusiastic and bubbly because we are now 12 days post-launch, and that was a long run-up to launch. So, of course, there's going to be a little bit of vibration here. So I'm an oceanographer. I work at NASA Goddard Space Flight Center, which is just north of Washington, D.C. That is the agency facility that is responsible for running this mission and has been involved for the full 10 years that it has actually been a real thing, but also part of the brainchild that led up to it 10 years before that. So a lot of the other enthusiasm comes from the fact that this is a mission that in the US is old enough to drink. It's been 2 decades in the making, and now we're 12 days past launch. So I'm an oceanographer by training. Recovered since I spend a lot of time thinking about flight projects now instead of oceanography, but I am still happy to be here. As for context, you have some wonderful data users to my left here. Just in case you're unfamiliar with the mission, it has 3 instruments on the full observatory. I wore this shirt because black is slimming, but also because it has a nice picture of the observatory on it so that I can point to things. The primary instrument is cleverly called the Ocean Color Instrument, which is our discipline. Basically, we're looking for the microscopic, often invisible universe of the upper ocean, but it's also highly capable for aerosols and clouds, and that was built at Goddard Space Flight Center as well. And then we have 2 small multi-angle polarimeters on the observatory. They were both contributions. The first was from the University of Maryland, Baltimore County, and it's called HARP-2. And the second is from a consortium of organizations in the Netherlands, and it's known as SPEX-1. So this trio together for the combined study of sea and sky is more information that on a global scale, on a daily global scale, than any other observatory ever flown has had, and there's nothing like it planned here or abroad. So in less than a week, the instruments will start their data collection mode, and then I think the real fun begins. So with that, I'm here for whatever questions you have, but you're more interested in these fine scientists, I'm sure.
Yeah, hi, I'm Clarissa Anderson, and I'm at Scripps Institution of Oceanography, as was mentioned. I'm a biological oceanographer, and as Jeremy mentioned, there's this thing called ocean color, and this is something that I've used a lot in my research to derive a better understanding of what's happening with the plankton in the ocean. And in particular, in my case, it's about which plankton are going to turn toxic and when. So all plankton, I say, are good, and sometimes they're bad. And every now and then we have to contend with those bad plankton that can bloom and cause a lot of harm to the ecosystem or public health. And so one of the things that's so special about the PACE, the instruments on PACE, is that it's going to allow us to do things like potentially detect the toxins themselves. Right now, we're very focused on how do we differentiate the plankton, but the idea that we could then go even further to understand the physiology, the stressors that affect these plankton, and then the secondary compounds that are produced is pretty fantastic. So one of the things that we've done in California is delve into taking satellite data, merging it with all kinds of other information to create what's called the California Harmful Algae Risk Mapping System. It's now an operational product at NOAA. And what it's doing is giving you, just like a weather map, a likelihood of encountering this toxin. This particular toxin is called domoic acid. It's a really nasty neurotoxin produced by a very adorable, sometimes friendly diatom called Pseudonychia. We're still not 100% sure when and why it turns on domoic acid, but when it does, leads to a lot of devastation. You're seeing here pictures of dead dolphins, dead sea lions, or sick sea lions. This is the case we saw a couple of years in a row recently. It's a really, really dramatic effect. It can also affect public health. When it gets into shellfish, you can get sick. It can also shut down things like the Dungeness crab fishery on the West Coast when there's a really bad bloom, and that affects people's livelihoods and the economy. So it has these cascading impacts. We have a really, really big interest in moving from some of the legacy satellite systems like MODIS and then VIIRS and into the PACE domain. So use PACE to be the workhorse for this model over the next 10 years or more as we transition to more robust models. Next slide, please. This is just an animation of that model. One of the things that we do at the Ocean Observing System that I manage is make sure that we're the connective tissue to the end users. We want the community to know these products exist, to know that they're driven by NASA produced data and information, but then figure out what do they need? Do they need a probability? Do they need that to be more granular? What could that look like relative to the resource that they manage? Is the resource shellfish? Is it marine mammals? Or is it really just that they're protecting local communities and livelihoods and need this kind of information to make daily decisions? This also affects policy. Next slide, please. And so lately what we've done is tried to connect the observations that we have in the ocean in California using these robotic microscopes called imaging flow cytobots to validate the kind of satellite data that PACE and legacy sensors are producing, and then to really fine tune and make next generation ecological forecast models of the plankton and how variable they are. Another thing we're doing is using a long-running fisheries program in California to validate the data that will come online for PACE. And this will be a whole range of interesting physics, biogeochemical, chemical, biological variables that we will be able to validate relative to PACE. And so along the lines of how all of this information comes together, the models, the satellite data to inform policy and decision-making, we're having a town hall here tomorrow called for the National HAB Observing Network. So it's the Harmful Algal Bloom Observing Network. And I just wanted to put in a plug for that so that you understand that this is an all hands on deck sort of situation. Thank you so much.
Yeah, so hi, I'm Shelley Tomlinson. I'm a research oceanographer with the National Center for Coastal Ocean Science. That is within the National Ocean Service of NOAA. I also wear sort of a second hat as the Coast Watch node manager for the East Coast. And what that is our satellite data information system office within NOAA tries to develop satellite products and makes them useful to, there's a lot of misunderstanding how to use the products, tools, trying to find the right tools to work with them. So we try to bring products to stakeholders on the ground, trying to work with the data. So my primary role within NCOS is in developing harmful algal bloom, like Clarissa had talked about, forecast systems around the country. So we develop satellite algorithms to detect different types of algal blooms and biomass, which is the concentration of the algae in the water. And then we use that to drive operational models to forecast when a species is going to show up at a beach. In the case, we have two operational models, one up in Lake Erie for freshwater harmful algal blooms, another in the Gulf of Mexico for a toxic bloom former called Corynea brevis. And what we do is we forecast when the bloom might hit the beach, or in some cases for freshwater, when it might get close to a drinking water facility, which is a huge issue with these toxins that cyanobacteria produce. And in the case of Florida, we have a respiratory model, which actually predicts when these toxins that get into the air, it can become aerosolized, get blown onto beaches. We predict on a beach level when those toxins might be at a particular beach to help beachgoers know when it's safe to go to beaches. So relative to PACE, we are currently using what's called multispectral data, which has just a short number of bands. So you can tease out where there might be a high amount of phytoplankton in the water, but you can't tease out what kind is in the water. So, you know, we use the ecology and the time of year to try and, you know, infer what it might be, but we really can't tell you what it is. So initially our plans are to take the PACE satellite data and create the same bands that we're using from other sensors so that we get another look during the day, because some of these we only get one shot during the day. So this will add additional information. So that's the first step. And then from there, we're going to try to take that data and tweak with this hyperspectral capability that you will have on that satellite, try and tweak those algorithms to be, you know, to get towards, can we now tell you that it's most likely a toxic organism versus non-toxic? So, and then, you know, incorporate that into our entire forecasting effort.
Awesome. Excellent. Well, I'm humbled to be with such amazing scientists. They're the best ones to be up here. But so today I'm actually going to talk about what we call the good phytoplankton. I'm here to talk about non-toxic phytoplankton, although knowing what the toxic phytoplankton is also super important. So I worked at the Ocean Ecology Laboratory for about 15 years. I've been an oceanographer for a little longer than that. I won't say how long, so you don't know my age. But I've loved oceanography. I've studied phytoplankton for many years, phytoplankton ecology. I've traveled all over the world on research cruises, collecting samples to understand phytoplankton and other optical properties of the oceans. But my heart lies in the polar regions, and it really always has since the first time I went to Antarctica. So for my dissertation, I focused on the Arctic, looking at the Chukchi Sea, which is the body of water between Alaska and Russia. And as you may or may not have heard, there's a lot of changes happening in these polar regions due to climate change. We're losing a lot of our multi-year ice. So ice is melting quickly, not as much sea ice. And sea ice is the driver really for all the ecosystems in the polar regions, really. And which starts with the phytoplankton, because the phytoplankton are the base of the food web. So understanding how climate change is impacting the food web, starting with phytoplankton, impacts everything up the chain to, you know, whales and walruses and things like that. So we really need to understand these changes that are occurring in the Arctic. And there are way more changes happening that I can't get into today, but there's a lot of things happening that we really need to understand. And actually the Chukchi Sea is on that right bottom panel there. And, you know, one thing we hope PACE will do, it should do, is because it's hyperspectral, it'll give us a lot more optical properties of the phytoplankton, which I'll show it in the next slide, not yet, but in a minute, to help us tease apart, as Shelley said, the different types of phytoplankton. And the reason I'm showing this slide is also because I'm mission applications lead for ISAT-2. One of the aspects that we're looking at is melt ponds. So in the top right panel is a picture that one of my colleagues took up in the Arctic. And those are melt ponds, which basically is melted water that pools on the ice, and it allows more light to penetrate the ice. So you can get more phytoplankton growing because they need light under that ice. So understanding those melt ponds, which is something actually ISAT-2 does, is also important to understanding the timing and magnitude of phytoplankton blooms in the polar regions. You can go to the next slide, please. Thank you. And of course, we can't, I know that some beautiful images of phytoplankton have already been shown, but these are actually images that were collected from colleague Sam Laney from Woods Hole, and these are actually from a cruise that I was on in the Chukchi Sea. So you can really see the diversity, variety, the shapes of all these different types of phytoplankton, and they all play a role in the ecosystem. And that top left picture of the different phytoplankton in bottles was from an experiment that we did looking at the optical properties of about 50 different phytoplankton species. So I like to include this because it shows not only are we looking at different shapes, but all those different colors that you see there, which is amazing. And we hope that, or we know, I should say, that PACE will be able to, pick up all of those different shapes and colors so that we can understand better the type of phytoplankton that are in the ocean. So thank you.
Thanks, everybody. And at this point, we'll open it up for any questions in the room.
Thank you so much. This is so cool. Congratulations on the launch after 2 decades. Oh my gosh. I'm James Dineen from New Scientist. Just to start, you talked about harmful algal blooms and different types of phytoplankton. What other types of things can we learn from ocean color?
Lots.
Yeah, I mean,
Yeah.
you can look at water quality. So, I work on the East Coast, Chesapeake Bay, they're always trying to manage the nutrients going into the water. So try and look at, temporal changes in water quality, like things like turbidity, water clarity, which is important for fisheries, for seagrass beds, et cetera. So that would be one case. What else? There's a lot.
There's a lot. I guess carbon export, right? So, I guess other people have developed algorithms to derive metrics of carbon in the ocean. So understanding carbon that's at the surface and what sinks to the bottom, which is also, again, part of that food web, and drawing things down from the surface to the bottom is also an important aspect, and PACE and other satellites help us to do that.
And if I can add, I often get asked, like, if you could pick a word or two that describes the mission. One of them is always interconnectedness, because we talk about this ocean color mission, and we're in an ocean sciences meeting, so you're getting the oceanographic perspective, but there is an immense capability for atmospheric retrievals from this mission. And that's important to the ocean sciences community as well. In particular, atmospheric aerosols, which are anything that's not a gas. So it could be pollen, volcanic ash, desert dust, pollution, whatever. And there are a lot of interactions between the sea and sky that are partially understood, but not super well on global scales. And so there are these phenomenon that we need a observatory like this to tie all the pieces together. Like, if you don't understand the ocean, you don't understand the atmosphere, you'll never understand the full system. And there are some cool stories that have come out even recently, like Australian wildfires in 2018 or so, where that actually seeded offshore in the South Pacific a major phytoplankton bloom, which offset some of the carbon from the wildfire. But there are also compounds that phytoplankton release that can help form cloud condensation nuclei. And so we're really in this to tie it all together. So just making sure that interconnectedness part was captured.
Yeah.
And what can you do from ocean color? That's why I was joking. It's a silly name for the instrument, because it's so much more than ocean color.
It is. And I think it's good, tagging on that is just talking about applications. And PACE has been a driver of this as well. Many NASA satellite missions are now incorporating applications, and applications meaning a societal benefit. So using the data for a societal benefit for non-scientists. So for example, monitoring for harmful algal blooms, looking at sea level change, all these things that, yes, we're very interested in the science, but we want them to be useful for non-scientists and decision makers so that they know, how to make policy to impact what changes that we're seeing now. So applications is hugely important as well.
And can I put a plug in too, since I'm in, no, there was one thing I forgot in my intro. I'm kind of in the operational side of all of this, right? So we're trying to make research sensors useful to, to groups, everywhere from public to state managers and stuff. But we're also using this as a precursor for an operational sensor. So you've heard of maybe the GOES satellites, the weather satellites. Well, in 2030 and beyond, we're really hoping to get a similar hyperspectral instrument on that 20-year series of GOES satellites on the East Coast, West Coast, looking at the US. So we're also using PACE to try and figure out how do we make these applications that you talked about useful, figure out how to use those hyperspectral bands so that when that launches as in an operational capacity, we'll be, ready to use it right away. So, and there is a town hall today if you're interested in that at 12:45
Well played.
called GeoXO.
Just to ask a follow-up about the cloud aspect, the atmospheric aspect that you often see, one of the largest uncertainties in our climate models is clouds.
Yep.
What's going to happen with clouds? Will this help get at some of those uncertainties and constrain
Yeah,
cloud
absolutely.
feedbacks with climate change?
In fact, one of the directives from NASA headquarters, way back 10 years ago was to produce an observatory that helped beat down the uncertainties, exactly what you're asking. And so this is where really those two multi-angle polarimeters come into play. Like they are supersonic detectors of microphysical properties of things in the atmosphere. And they do this in two ways. The multi-angle part of it is that whatever piece of real estate they're looking at as they fly over, they're seeing it from different view angles. So that has a lot of power in terms of shape, height, thicknesses. But they also are polarimeters. So envision polarized sunglasses, like you're just seeing the universe in a whole different way. And when you put that together, the novelty of some of the retrieval space is grown to become immense. And so cloud bows, which are like rainbows made by clouds, are retrieval from this, aerosols as well. And now that you have those measured together from these newer instruments with the ocean biology, those uncertainties should collapse.
And I might add to that in concert with PACE, there will be an important airborne campaign called PACE PACS. And this will have similar analogous instruments with these polarimeters, but also ocean color instruments or instruments that span a really wide range of the spectrum that will look at resolve clouds, resolve the ocean, but at a very different height elevation in the atmosphere. And those two coupled together give you a lot of power because you get this synoptic look from the satellite, but then you eliminate a lot of the atmospheric problems when you do these airborne campaigns. And I also think blending those together is going to be extremely powerful for resolving clouds, but correcting for the atmosphere that often interferes with our ability to look at plankton.
It's the equivalent of the ground truthing
Mhm.
that you're doing with
Call
the
it sea
the plank-
truthing. Yeah.
sea truth. That's better. Yeah.
Hi, oh, can you hear me? Okay, hi, my name's Evelyn McDonald. I'm both the journalist representing Random Lengths News in San Pedro, California, and a professor at Loyola Marymount University. So thank you for being here. This is very fascinating. Usually on Tuesday mornings, I actually volunteer in animal care at the Marine Mammal Care Center in San Pedro. So I helped deal with the incredible influx of animals we had last summer. It was just really, really, really sad. So I saw my first, dead baby dolphin come in and things like that. So this is impactful on different levels. So one of my questions for you, so what is the mechanism by which this information will get relayed to journalists, to the public, to places like the Marine Mammal Care Center knowing in advance that this might be happening would have been tremendously helpful for us there to staff up and also just, how are we going to find out when these blooms are coming?
Shall I take that? Yeah, so the model that I referred to, California Harmful Algae Risk Model, it did capture this event. The reality is that we're often, all of us, understaffed enough to then call up all the marine mammal centers and say, look, we think we see something. There can be false positives. We don't want to have everybody staffing up in a case where it might not pan out. But as soon as it looked like it was a real event within 1 week, my organization, we were working very closely with your group and all of the marine mammal care centers in California to deliver, day by day risk maps, data. So there is a conversation happening. That pipeline can be vastly improved, and we've been looking into how to do that over the last year, particularly because of these two incredibly impactful events. But we can do better. The problem with forecasts is always going to be that we're pretty far, even with powerful satellites, away from seasonal forecasts. We are looking at things in that weather scale domain, 3 days, 7 days out. It is hard to prepare, and that's the only kind of time horizon you can really learn this from. But I think that as we get these hyperspectral data, I think we can not only refine the detection part, which is more of a real-time detection, but we can start to feed some of the more next generation forecast models that we have with validation data that will improve those seasonal to even multi-year forecasts of risk. And we're really hoping that if we can refine that accuracy, then we won't be hit completely off guard. But we're also contending with climate change, and we're contending with some pretty nonlinear shifts that we're having a hard time predicting in general.
Weather in general. Well, and then correct me if I'm wrong, but with marine mammals too, like it takes time for them to accumulate the toxins and then get sick. So the timing of when we see a bloom versus when it actually is sick and dying at a beach.
It can be 1 week.
Weekly? Okay. Yeah.
Any other questions in the room?
Using this word hyperspectral and comparing that to what exists now with the multi-spectral instruments, can you sort of quantify how much of a better view PACE gives us on color here? Yeah. To put some numbers to that.
Yeah, that's great.
Where are the grants?
So I mean, the nice thing about Ocean Color is that we're standing on the shoulders of some giants here. So there are other NASA assets, and it's acronym soup. I'm sorry, I'll expand anyone you want, but MODIS and VIIRS you've heard us talk about. And there are European instruments that do the same, Asian as well. They basically operate by just measuring several colors of the rainbow in the order of, let's say, 5 to 7, depending on what they are. So in principle, the instruments operate just like your eyes do. You interpret color into what you think might be there. So if you're looking at a blue body of water, you would intuitively say, hey, this is pretty clean. If it's green water, biology is happening, brown water, suspended sediments, or organic material. So the bands that exist on our existing satellites are intelligently placed such that they can pull a phytoplankton signal out from everything else. And that's great because today we can say we see where the phytoplankton are, and we can give you a good estimate of how the abundance is. But like if you've ever walked through a forest and just looked at all of the, like we measure two bands of green, but there are all kinds of greens. And your eyes are telling you, hey, I can see differences in this plant versus this plant just by, hey, this is a different color green. So today we've got the box of crayons, that's 8. And where we're going with this hyperspectral is a continuous spectrum of light that runs from the ultraviolet all the way into the near infrared. So the crayon analogy is now we have 200. So we're seeing all the greens. And as you saw from Amy's slide with the cultures from the laboratory, there are a lot of different colors out there, not just 5 to 7. And so this progression of the rainbow that we can collect now is really that massive innovation. And there are other instruments that do this, but none that do it on a daily scale. And that's really where we're coming in. So yeah, boiling it down, if I said, give me the best picture you can ever draw, you're going to take the box of 8, you're going to take the box of 200.
Other questions?
Congratulations. It's just an amazing leap forward. How long is it going to take you to see through all that color range to when you can get to what Clarissa is trying to do in terms of getting a useful forecast?
So it's kind of that's a religious question in a way. So the good news is that there are instruments that we have in the laboratory and in the field that we can take on ships or towers, or like you were mentioning, airplanes, that mimic what the satellite will measure. And so we do have the sea truth from instruments we have in hand here. But that said, the world is very diverse and it's changing, and there is no one series of measurements that could represent all places at all times. So right away, we will be able to be some places collecting data that we can use as sea truth, but in terms of nailing down like a really statistically meaningful analysis, well, the world's a big place. There's a lot of ocean out there. It's changing all the time. Phytoplankton aren't like land plants. If you don't see them today, may not be there tomorrow. So fortunately, we have funded programs that will run at least 4 years, where there's no hypothesis. It is simply NASA paying PIs around the US with some international partners to collect as much data with as much diversity as possible. But that said, you know, hey, there are instruments on orbit that have been there for 20 years. We're still assessing their performance.
And if I may add to that, one of the things that I've learned a lot is we can be really practical with data we have now versus, you know, really what are we going to develop with this new capability? For instance, I mentioned this model we have, it's built off of the multispectral capabilities of the past. So when we start to transition it into using PACE information, we're still just going to be cherry picking those few bands that it was created from, you know, when we had a multispectral sensor. So that is a very practical way to take something that is working for the community and continue it with PACE information. But then you need to take the hyperspectral information and start to think, what does the next 10 to 20 years of harmful algal bloom or plankton modeling and detection look like? And these are things that are going to be happening in parallel. So there's the here and now, and then there's, you know, the vision.
I guess I could add one more thing to that too, as far as prepping for using the hyperspectral. So we also have a lab in Beaufort, North Carolina, and I know there are others around the country, researchers doing the same thing, but in trying to tease out the spectra or the color, you know, spectra for different phytoplankton in a lab setting. So controlled, you have a culture in a beaker, and we're trying to figure out what does that look like with none of the other crud in the water, right? So then the next step would be, all right, now how does that look from like a drone or aircraft, and then eventually translate to satellite? So there is stuff going on in the lab to try and figure out what's that suite of algorithms we should be using from PACE. So TBD on that. I know she's had real success with cyanobacteria because their structures are very different. They clump into, you know, bunches and things, so it's easier to tease out than some of the, like the dinoflagellates and things that I work with, but we're hopeful.
With the current multispectral instruments, are there species of with the current multispectral instruments, are there species of phytoplankton that we're just fully not seeing, or is it that all phytoplankton are just sort of put in this one, you know,
No.
not very resolved green band?
I mean, we could talk about that for hours, of course. There's a lot we're missing. One of the things that I'm really excited about with PACE is that we're going to be getting down into UV channels, ultraviolet channels, like getting all the way to 380 nanometers that we've never been able to do with some of these legacy sensors. And one thing we really do know is that some of the compounds, protective compounds that some plankton make, are highly detectable in those channels, but those plankton are also some of the ones that can turn bad. And so one of the ways to think about how we can capture those plankton would be to use those UV channels. Often you need to use them against some other, anchor it to some other waveband. But we barely, I think, are scratching the surface on what that capability looks like. We know that we can use that channel. We've done it with other hyperspectral or UV sensors that are in space right now. But this will really, I think, change that landscape. But there's so many ways in which we're missing shifts in color. We talk about red tides a lot, often missing key shifts in that sort of that orange, yellow to red shift part of the spectrum. We've been missing that for years in multispectral sensors, and that's really going to be critical.
Yeah, and another example with the cyanobacteria that I mentioned in Lake Erie, most of the time we can pick it up very, very well. But often in late spring, there's what's called a chlorophyte. It's a green bloom that shows up that sometimes can get triggered by our algorithm when we're like, we know it's not the right time of the year. It's not the toxic thing. But again, they do pick up other things. It's just really hard to separate. So we're really hopeful that this hyperspectral capability will help with that.
And the other thing about phytoplankton from that picture I had up earlier, you saw the different colors. Not to get too sciencey, but those colors are caused by these compounds called pigments in the phytoplankton cells that harvest light. So we use those colors to kind of differentiate different types of the phytoplankton, but a lot of them have similar, they could be very different types of phytoplankton, but they have similar colors because they have the same of those pigments. So with the hyperspectral, we can get more of those colors out of the phytoplankton, which would be phenomenal, and also get more information about the size and the structure of those cells with other measurements to really further tease apart the different types of phytoplankton that are looks sort of closely together color-wise, but are so very different. So I think PACE is really going to help that with that as well. So we're all very excited about it, if you can't tell. So if from the last slide where we were showing the different phytoplankton images, I mean, some, I don't know, some phytoplankton can be teeny tiny that you need a microscope to see, and others can be quite a bit bigger, form these really huge chains that sometimes you can see with the naked eye if they're big enough. So especially diatoms, which are phytoplankton that have a silica shell essentially, and they create all these amazing, beautiful shapes. So all of those different structures, the chains, they have spines, whatever that morphological characteristic is, also helps us define what the different types of phytoplankton are.
I guess what I'm saying.
Yeah.
The satellite captured the size of
Well,
micro.
in a way. So basically, and Jeremy can jump in if I go down the rabbit hole of this. But basically there's light can bounce off of things, right, in the ocean. So the way that the light bounces off of these particles in the ocean, these phytoplankton, we can use that information to actually derive or compute what the size of the phytoplankton are. So it's kind of that combination of color and that other signature to get at the size and morphology of the cells that we can put together to determine what type of phytoplankton it is.
Or
Nice job.
whether it has the silica or it
Yes,
doesn't either, right? Like
exactly.
the outside of the cells can be very different.
Yes.
There's spiky ones, there's, blobby kind of, so,
Yeah.
and they all reflect the light differently.
Mhm.
It's called backscatter, but
Yeah.
the way they scatter the light in the water, it can really vary.
Mhm.
We're talking about huge differences in size, 5 microns to 200 microns.
Yeah,
Mm.
really, really.
To us, these
Big
are big
difference.
differences, but it's rather comical when you think about
Yeah,
it.
yeah.
So I may have misheard, but I think in the beginning, you said you could pick up when the species turns toxic. Is that? So is
Yeah.
it the same kind of thing? Like when they
Yeah.
turn toxic, they change what they're backscattering or they're something. So
Well,
how would you
Yeah.
would you do that?
We have evidence that, Shelley was alluding to this earlier, that you have, you're getting spectra from ocean color measurements. Those spectra have shapes. So when we look at those shapes, blooms that are very toxic often have a very different spectral shape. Why? We're not entirely sure. It could be because of some sort of secondary change in the ecosystem or in the water or in minerals or in calcification or silicification that we're detecting, or in pigments or in, secondary pigments, but it seems to be connected to the production of the toxin. So I don't know that we'll ever really be detecting the chemical signature of the toxin. But I think there's a lot of power in resolving these shapes in a way we couldn't resolve them before. That tells us about the physiology of the organism. We're already learning about that with other instrumentation. Soon we may have LiDAR that can tell us things about stress that gets us all the way to what is iron doing in environments? Iron turning on stress in plankton. So we're sort of inching our way towards more inference about the changes and conditions in the ocean that lead to changes in plankton that lead to cycling of nitrogen, carbon, and other really important biogeochemical variables.
Any other questions? All right. Well, thank you so much for coming to our panel, and stay tuned for our first sets of data from PACE coming soon.
Yeah, thank you so much.
Okay.
Thanks for the
Yeah,
interest.
thank you.
Yeah, thank you.
So this week is. The instruments have been powered on. They'll start collecting engineering data at the second half of this week. And then first scientific light will be roughly 4 weeks from now. In which case the floodgates will open.
I'm terrified.
Thanks so much, PACE panel. The recording from this session will be available this afternoon to registered reporters on the Press Information Exchange, along with the slides that you saw. We'll also post this to YouTube this afternoon. We'll make that link available to everyone. Thanks for coming. We hope you come back at 2 this afternoon to learn more about ocean-based carbon dioxide removal promises, risks, etc.
Oh, cool.
Thanks everyone for coming.
So they're doing these.
Thank you all.
Sounds like it's pretty cool. I had no idea. I didn't either.
Yeah, you're welcome.